Assalamualaikum warahmatullahi wabarakatuh. My name is NUR RAIHANAH BINTI JAAFAR, and I will present about the determination of species diversity, the insights in the rim of Nepenthes species, and abiotic factors that influence the Nepenthes diversity in Cameron Highlands, Malaysia. During the discussion, we will go through the introduction, literature review, methods, result, discussion and conclusion, and recommendations. Nepenthes, commonly known as tropical pitcher plants or monkey carps, are a group of unique and fascinating carnivorous plants that play important ecological roles in their native habitats. However, despite their ecological significance and captivating features, Nepenthes face several critical challenges that threaten their diversity, distribution, and long-term survival. Nepenthes, commonly known as tropical pitcher plants or monkey carps, the first objective is to determine the diversity and abundance of Nepenthes gnats to identify the types of insect that thrive inside the Nepenthes and to determine the abiotic factors that influence the Nepenthes diversity and abundance at Mentigi Hill and Mountain Irau, Cameron Highlands. Through this study, the researcher may find the new species of Nepenthes and analyze the abundance of Nepenthes at the selected locations. In addition, the analysis that will be presented in this study will give valuable and crucial information for future research for conservation of endangered or valuable species of Nepenthes that based on the abiotic factors. Lastly, the findings of insect that thrive in the ring of Nepenthes feature will give information about the type of insect that attract to the Nepenthes. The two study areas for the study is Mentigi Hill and Mountain Irau. Mentigi Hill is a mountain located in Cameron Highland and has elevation of 1535 meters and situated near to Tanah Rata. The other location is Mountain Irau, and it is the Malaysia's 15th highest mountain with 2110 meters. The mountain Irau's wonders include mossy forest and fairyland, cold wine, and misty cloud. Species diversity is defined as the total number of species and the population species in a particular ecosystem or environment. Species diversity is one of the most commonly measured quantities in ecology, but sometimes it is complicated. Species abundance refers to how evenly individuals are distributed among the species within a community. Species abundance is the number of individuals per species. Despite having same species richness, two communities may have different relative abundance. Species abundance provide valuable information about the distribution and dominance of species in the particular ecosystem. The Nepenthes plant is a semi-woody vine or scramble that cannot stand upright without external support. The distinctness of Nepenthes lies in the variety of its feature, which can come in variety of shapes, size, colors, and patterns. Most Nepenthes species have two feature types that reflect the plant's developmental stage. Lower or rosette pitchers are typically produced by immature non-climbing plants or basal side shoots on stems with short internodes. Upper or aerial pitchers are produced by mature beanie plants and are frequently born on prehensile tendencies that both position that pitches in more exposed in microenvironments and facilitate the plant's climbing habit. The use in this study was plotting methods. Plot sampling is a method of estimating abundance in ecology in which plots are chosen and sampled from within a survey region. The method of gathering information involved determining location of the research mapping and mapping plot transit lines until it was recovered, gathering description of morphological features of Nepenthes and the body structure of insects trapped inside, and conducting a list and verification. A total of 5 plots were set up at each of the sampling site. The size of the plotting was 20 meters times 25 meters. The site of the sampling are randomly chosen. Each stand should have at least 3 total plots. This may mean that the plots near together in smaller stands, but they must never overlap. There are three species of Nepenthes that can be found in Mentigi Hill and Mountain Irau, which are Nepenthes maffelni, Nepenthes sanguinea, and Nepenthes gracilis. Nepenthes maffelni is a widespread endemic and distributed on certain mountain peaks located at the central and western part of Peninsular Malaysia and prefers upper mountain forest ridges. It is locally abundant in its range, although it has distinction in distribution. Nepenthes sanguinea found in southern Thailand and Peninsular Malaysia at elevation ranging from 300 to 1,800 meters above sea level. This implies that it covers lowland, intermediate, and highland Nepenthes territory. Nepenthes sanguinea pitches range in color from pure green to blood crimson to reddish brown. The interior of the pitcher is often pale green and mottled red. The short, elongated pitches of Nepenthes gracilis are unimpressive with a very thin peristome. Nonetheless, the species is uncommon in that the pitcher lid has an uneven covering of wax crystal on the bottom. This layer is not as thick as and structurally separate from the found in the pitcher's wet sea zone, and insect can easily stick to it under dry conditions. In the first plot, Nepenthes maffelni and Nepenthes gracilis were found. The total number of individuals for Nepenthes maffelni are 5, while 4 for Nepenthes gracilis. In plot 2 and plot 3, only Nepenthes maffelni was found with 8 and 15 individuals respectively. In plot 4, Nepenthes maffelni and Nepenthes sanguinea were found with 25 and 30 number of individuals respectively. Lastly, in plot 5, only Nepenthes sanguinea was found with 36 of individuals. Environmental preference of Nepenthes species vary in addition to the diversity in feature form. Others are adapted to highland forests, mossy wood atrophic environment or pet swamp ecosystems, while some species thrive in lowland rainforests. The total species richness is due to the diversity of habitats. The Shannon Index were used to calculate the species diversity of Nepenthes at both locations. The PARS version 4.03 software were used to analyze the data. The value of Shannon Index is 0.8082. The percentage that each species make up of the overall population is referred to the species evenness. When calculating the Shannon Wilderness Species Diversity Index, the number of each species and its percentage of the total numbers of individuals and multiplies each species fraction by its respective natural log. The graph sloping shape that indicate that data of species and even and diverse and the number of sampling is enough. A refraction graph is a graphical tool used in ecology to evaluate and contrast the richness or diversity of species found in various samples or communities. In a refraction graph, the y-axis indicates the estimated species richness or diversity, while the x-axis represents the number of individuals in current samples. The graph includes a line or curve showing the estimated species richness after refraction in addition to plotting the observed species richness as a function of the sample size. Species abundance can vary significantly and is impacted by a number of variables such as environmental conditions, ecological interaction, and species-specific traits. Nepenthes maffelni and sanguinea showed the high abundance, and Nepenthes maffelni was found in almost all of the plots, especially in plot 3 and 4. Nepenthes maffelni is known to be the most common Nepenthes species in Malaysia regardless of the soil condition. Based on the past version 4.03, the evenness is 0.748. The broken stick model suggests that species abundance are dispersed randomly. It makes the supposition that the distribution of scarce resource among species at random abundance that are more evenly distributed. The broken stick distribution is frequently seen in areas where there are few ecological interaction or when there is a fair amount of resource availability for all species. Insect that trap inside the pitcher plant all can be found in the plot at Mount Irau and Mentigi Hill. It consists of two orders, which is Diptera and Hymenoptera. The other Diptera including blackfly, Parasimulium furcatum, and midges, deceit midge. Diptera have three pairs of legs, one of each of the three segments of the thorax, which are known as the fore, mid, and hind leg. Diptera larvae are apodous, but some particularly aquatic larvae have appendages comparable to Pseudopodia. Ants, bees, wasps, and sawflies are all members of the enormous and varied order Hymenoptera. A few distinctive characteristics define Hymenopterans. Order Hymenoptera go through complete metamorphosis, therefore their life cycle includes separate egg, larva, pupal, and adult phases. Nepenthes species require particular temperatures for optimum growth and development. Typically, they can be found in warm to subtropical setting. The temperature can have an impact on the general fitness and distribution of Nepenthes species by affecting a number of physiological activities such as photosynthesis, nutrition intake, and metabolism. Specific thermal niches may be occupied only by species with limited temperature tolerance, leading to localized patterns of diversity. Heat stress has been demonstrated to alter plant cellular structures and defense mechanism, enhance respiration, decrease photosynthesis, and increase the generation of stress metabolites in plants. Nepenthes can be divided into two groups: lowlanders, which can withstand heat stress and are found in hot, humid jungles below 1,000 meters above sea level, and highlanders, which are found in highland mountain forests with warm winter and cold to cold, humid nights that can withstand cold stress. In conclusion, the genus Nepenthes is remarkably diverse and abundant. They inhabit a variety of habitats primarily in Southeast Asia, including lowland rainforests, highland forests, mossy forests, and other specialized settings. Geographical location, climate, habitat variety, soil quality, disturbance regimes, adaptations, and human activities are some of the elements affecting Nepenthes diversity.